Well, welcome. This is John Bagnulo, Director of Nutrition for Functional Formularies, and I'm glad to have you with us this morning to discuss the role of inflammation in the immune response and in chronic immune-based conditions and diseases. It's an area of nutritional science that expands rapidly every week as we learn more and more about a variety of sources of inflammation that are in constant communication with our physiology. So with that being said, I want to get going here as soon as possible. We have a lot of ground to cover. By now, I'm sure that many of you have had at least some exposure to clinical papers or to other sources of scientific information regarding the role that the microbiome or the GI environment plays in governing inflammation. And that's certainly going to be the predominant source of inflammation for the human body. And of course, there are other sources, whether we talk about the role of acute or chronic injury or infection is certainly playing pivotal roles in the inflammatory response and in how that inflammatory response is interpreted by the immune system. But really the gut plays such a pivotal role for many reasons. And the primary reasons are that you have this enormously complex ecosystem there with anywhere from 300 to, in a very diverse intestinal environment, 1,200 maybe species of gram-negative and gram-positive bacteria, plus eukaryotic species such as fungi or yeast and viruses, and in some cases, parasites that make up this microbiome. And that mix of species has an enormous influence on what happens at the gut lining. Then you also have, of course, the importance of particular components of our diet that may either feed families of bacteria, feeding one more than others. You will have other dietary components that may be additives to food, chemicals, agrochemicals that are used in the growing of food, and then other substances that may be produced during food processing or heating. And all of these create an incredibly large number of moving parts that can influence that intestinal lining and all of the different receptors that lie in close proximity to that intestinal lining that can pick up on either potential threats or ultimately sources of inflammation. Now, it's really important to note that a large percentage of our immune system, in some cases, depending on the individual, but in some cases, 3/4 or 75% of the immune system lies within just a couple centimeters of the intestinal lining. That's really important when we think about all of the different ways that diet and that microbiome can influence the immune system's activity and consequently inflammation. Another important aspect to note is that a high concentration of particular families of leukocytes, for instance, eosinophils, which can have very aggressive defense behavior patterns when it is interpreted that there's a threat present, they're also right there at that front line. And then, to build upon this, the intestinal lining, just as a kind of an overview, has over the course of human evolution, it has been given a variety of gateway or portal type of regulatory exposures to what's in the GI, meaning that at times these windows will open and allow our immune system to have some short windows of exposure to potential pathogens, or pathogens for that matter, and other possible threats that the immune system will have to produce antibodies to for later defense against. And that's a big part of this mechanism for immune programming that over the course of human evolution really evolved to have acute exposures, not chronic exposures to particular aspects of our diet or particular species of microbes. Now, again, as an overview to all of this, a chronic and sometimes acutely intense stimulation of particular families of leukocytes that are there at that intestinal lining can generate an overly aggressive immune response or an immune-based cascade of events that ultimately contribute to the pathology of one or more tissue types. That may be connective tissue, it could be nervous tissue, and there are countless examples of that we'll take a look at in a moment. But here are really the leading causes of diet-based, food-based inflammation. If I had to narrow it down to the 5 most common sources, the first would be dysbiosis. I feel like there is an overwhelming body of clinical evidence now showing very strong relationships, and in some cases, cause and effect relationships between particular pathogenic bacteria, families of bacteria, imbalances, and inflammation or autoimmune conditions. Then there are also a significant number of papers, which this is quite often novel information for clinicians, that show the damaging effects of lectins found in a wide variety of plants, most notably legumes in certain grains, that damage the gut lining and really increase permeability. Then there are, of course, non-compatible proteins, which are in the headlines daily, and most people are now familiar with the damage that casein or gliadin or something like soy protein has been shown to cause. And again, researchers have found now various mechanisms between these less compatible proteins, whether or not our immune system is interpreting them as a foreign invader, once they're in circulation, whether it's the role of something like gliadin, which is the most common type of gluten, in stimulating zonulin production and thereby increasing intestinal permeability. There's a wide variety of mechanisms that can explain the inflammatory effects of these less compatible or non-compatible proteins, but they're something that most of us have had, again, a certain amount of exposure to. Then there are the roles of less naturally occurring and more either synthesized or food processing generated compounds. These would be 2 major categories. We'd have advanced glycosylated end products, sometimes referred to as age molecules. These are produced in high heat cooking. And then you have a variety of artificial ingredients and chemicals, some of which are used in clinical trials when they want to generate high levels of intestinal permeability in an animal model. They actually use specific additives or emulsifiers that are used in the food supply, which is really, it's really puzzling when you start to think about, that paradox that we're using it in the food supply for a particular aspect of food science, and yet we're also using it in animal models to generate an accelerated level of GI pathology. So these would be the 5 most common sources of dietary-based inflammation. But again, dysbiosis would be at the top of the list, primarily because it can go in so many different directions. Now, in the past month or so, there have been a handful of papers. I put these references at the bottom of this slide, if you want to look at these more closely, that have been looked at by many gastroenterologists, immunologists, registered dietitians, and a host of clinicians as major breakthroughs. And they're really just, I would say, more recent affirmations of prior research which has shown very, very similar results, but in different models or in different time frames. What these last couple of interventions or trials have shown is that there are at least 4 different ways in which dietary modification can influence immune function, autoimmunity, and overall levels of chronic inflammation. Those would be dietary restrictions and or fasting. It's been shown to stop a deteriorating immune system and actually improve an immune system's ability to recognize friend or foe and to be more effective. Fasting has also been shown to promote a highly anti-inflammatory, more systemic type of process throughout the body. Fasting has also been shown to alleviate and in some cases reverse autoimmune diseases. And a restricted diet with respect to eliminating lectins, eliminating those dietary sources of less compatible proteins, which we just talked about on the previous slide. Whether we want to call that a specific carbohydrate diet, a more paleo diet, a gluten, dairy, soy-free diet. There are a lot of different ways with which we could look at that particular dietary intervention. But overall, these dietary restrictions have been shown to significantly improve outcomes with ulcerative colitis and Crohn's disease. And at that most recent trial, which was directed by Dr. David Suskind at the Seattle Children's Hospital, in a very short period of time, children with ulcerative colitis and Crohn's disease had all symptoms mitigated or basically were put into remission with this carbohydrate-specific diet. So these are, for some of us, these are really eye-opening revelations that, we may have had in the last month or two, but these are just added to a growing body of clinical evidence that dietary restriction and/or dietary elimination, getting rid of the most problematic aspects of our food supply, can have huge or produce huge improvements on outcomes for a wide variety of autoimmune diseases. Now, I've given you a couple examples of, where immunity can be overly upregulated, and there are just an infinite number of combinations. But if we're looking at something, as in the case of type 1 diabetes, it's well established that a significant, if not majority, of cases of type 1 diabetes are really where you have autoimmune damage to the beta cells, and it's been well established again that inflammation and destruction of the beta cells can be driven by chronic exposure to gliadin, again, a type of gluten. It can be also driven by particular patterns of dysbiosis, and similar patterns of dysbiosis, if not driving type 1 diabetes, have also been linked to rheumatoid arthritis, and again, other autoimmune conditions that may be affecting respiratory function or nervous function. But systemically, dysbiosis has the ability to influence autoimmunity and to deregulate immune function. When it comes to heart disease, there is also really good evidence that leukocyte adhesion, which is an important part of the etiology of cardiovascular disease, having that leukocyte adhesion, having the oxidized low-density lipoproteins infiltrate the or the artery lining. This is accelerated by leukocyte adhesion, and those leukocytes, or those white blood cells, become much more likely to adhere when they are inflamed, and they're upregulated by a variety of substances that may be either detected at the gut lining or may actually be in systemic circulation. Then, of course, you have substances such as the lipopolysaccharides that are produced by specific, or generated by specific, I should say, microbial or bacterial populations. And they can activate these lipopolysaccharides produced by primarily gram-negative bacteria, can activate a white blood cell and can really increase that, because of the inflammation that will occur when that white blood cell is activated, can significantly increase the permeability of the blood-brain barrier. This is going to cause problems with neuron health, and it can cause microglial type developments. And when I say certain microbial populations have the ability to generate these lipopolysaccharides, they're predominantly gram-negative bacteria, and some examples of gram-negative bacteria, to take you back to cell biology or microbiology, many of these, not all of them, some are benign or commensal bacteria, but many of the more problematic pathogens are gram-negative, such as the Shigella producing, toxin-producing gram-negative bacteria, Salmonella. Klebsiella is one that I have always had on my radar when working with people clinically, because Klebsiella is really implicated, or Klebsiella overgrowth, I should say, is implicated in a host of autoimmune conditions because of how unique it is in producing a variety of substances that are often misinterpreted by our immune system. And I think, using Klebsiella pneumonia as an example of where a specific type of dysbiosis or the presence of a pathogen can initiate inflammation and autoimmunity. It's one of the best examples I can give. It's really fascinating. So Klebsiella is something that many of us carry in at least small numbers within our GI. What the percentage or the population of a Klebsiella family of bacteria has to get to where it's an issue is it's an important question. I don't think we know definitively yet, but certainly higher levels of Klebsiella are a risk factor for a variety of autoimmune diseases, and this is really how it unfolds. Klebsiella, in response to dietary starch, produces an enzyme that helps it digest the starch. And that enzyme, this is a mouthful, but it's called a pullulinase. And a pullulinase produced by this family of bacteria helps digest highly branched starches, which we call amylopectin. Now, this pullulinase, which is an enzyme, is in contact with our brushboard or our intestinal lining. Unfortunately, it has a very similar amino acid sequence to a variety of collagen types in the human body. And what happens is when that pullulinase reaches the brush lining and it's in constant exposure, the immune system, and you can see on this slide that the variety of pullinases are used to degrade this amylopectin into smaller glucose chains or smaller oligosaccharides. That pullulinase is interpreted by our immune system eventually as a pathogen. And because Klebsiella is chronically producing this enzyme to digest the starch in our diet, and we're feeding the Klebsiella population by eating significant quantities of dietary starch, then we have an overproduction of this enzyme. It is interpreted by the immune system as a threat or pathogenic invader. And then eventually, because of its similarity to the protein code that makes up the enzyme and the protein code of our collagen, eventually our immune system starts to attack our own collagen. And this is, in essence, ankylosing spondylitis, a debilitating autoimmune disease that is growing in terms of its incidence. And one of the most effective ways to mitigate symptoms or to treat this condition is by placing an individual with ankylosing spondylitis on the appropriate antibiotic-based treatment and a low starch, or in particular, a low amylopectin containing diet. And that would be, for instance, white potatoes. They're a very rich source of amylopectin as opposed to amylose, which could be found in higher concentrations in, let's say, rice. So this is just one example of a particular type of dysbiosis that is found when we have Klebsiella overgrowth. But there are, yes.
Can I just interrupt? When your mic is a bit scratchy, is there any adjustment you can make causing background noise?
Let me unplug my mic. Is that any better?
I believe so. Yeah, that took away the scratching.
All right, thank you.
Thank you.
So these different families of gram-negative bacteria that overgrow or become a real dominant force within the GI, those are sources of dysbiosis. And ultimately it's just one example. There are others, such as the Shigella bacteria and Crohn's disease as well, Citrobacter and ulcerative colitis. And really the keys to understanding or addressing these are to recognize the sources of flora as an imbalance and overgrowth, as that's where the inflammation is really coming from. And so correcting this may involve antibiotic therapy. It may be other substances may be used as well to shift populations, whether it be botanical oils or other things. But ultimately, it's really important to consider dietary manipulation, because that's what will affect the metabolism of some of these pathogens. And whether Klebsiella might require a low amylopectin or a low starch diet, sometimes referred to as the London diet, But if someone had Shigella overgrowth, I would recommend a diet that was very low in the sialic acid NEU 5GC. Now that diet would be really low in red meat or no red meat. It would be very low in dairy, maybe no dairy, but would contain eggs and fish, which are basically free of that form of sialic acid NEU 5GC. And the same might be true of Hashimoto's as well, because certain metabolites are much more important for particular families of bacteria that have become pathogenic. And we'll try to leave a good amount of time for questions towards the end of this. So the next area where dietary factors can drive inflammation would look at the role of substances that influence one of these gatekeepers to that level of intestinal permeability, or those tight junctions. One of those gatekeepers is referred to as an inflammasome. And an inflammasome is really, the best way to describe it is, it's a complex structure of proteins that regulate both gene expression and protein production within a cell. And these inflammasomes are in almost all cells, and they really influence what happens at the surface of the cell, what's produced for proteins that, in the case of the gut lining, can really open or close doorways and can produce substances that are going to trigger inflammatory cascades, some of the interleukins, for instance. Inflammasomes are in constant contact with every aspect of our diet. Every ingredient, every molecule within our diet ultimately has the ability to upregulate or downregulate this inflammasome activity. And when we look at some of the more potent examples of inflammasome stimulating substances, they are also quite often highly interactive with another important gatekeeper of the intestinal lining, which are called inducible heat shock proteins. And the abbreviation on this slide would be the IHSP. So an inducible heat shock protein is really a protein that also controls the entry of substances between cells. In the case of the intestinal lining, they can help repair damage and some of the opening or permeability that may have been generated by something like zonulin. So these inducible heat shock proteins, coupled with the overall inflammasome response within a cell, together play pivotal roles as gatekeepers. They're going to keep substances out of the blood. They're going to keep substances out of circulation that can drive inflammation to high or condition-producing, symptom-producing, or disease-causing levels. So these are two really important mechanisms to understand. And some of the substances that are very potent inflammasome and inducible heat shock protein altering ingredients are found in a wide variety of plants, and they're called lectins. Now, lectins ultimately are produced by plants in an effort to defend themselves against a variety of pests. Those pests may be insects, they may be herbivores that are grazing on a particular food, but lectins defend a plant. In the area of botany, they are the primary defense mechanism used by plants against a variety of pressures within their ecosystem. Most lectins have an ability to make either the red blood cells or the white blood cells, for instance in an animal, agglutinate or form these clumps. And again, I show you this slide from the reference that you see at the bottom of the slide so that it becomes clear that there are certain sources of dietary lectins which are much more well studied and well understood than others. And on this slide you see Phaseolus vulgaris, that's the kidney bean. Another example which is Dolichos biflorus, it's the third one down, that's the cowpea. These are very common legumes that are consumed, typically infrequently by most populations in the world, but some people develop dietary habits or practices where they eat one of these things quite regularly, and they have adverse effects on people. Sometimes it's much more so in one individual than the other, but these are prime examples of leukoagglutinators, and in the case of the cowpea, a hemoagglutinator. They really cause problems for white and red blood cells. They're considered toxins in the field of toxicology, and for people that want to dive into this deeper, there are some references I can share with you again towards the end of this talk. But lectins are, even though they're naturally occurring, even though they are part of a plant-based diet, can be significant sources of inflammation because of the way they affect hemodynamics, or the way they affect the gut lining. And really, to clearly understand them, it's just important to know that something like the cowpea, for instance, the lectin that has been isolated from the cowpea, and this is true of other legumes, these have been used for decades as methods. Once they're isolated, they've been used as methods of selecting, or I should say screening, for certain blood types. So when labs are trying to look for certain patterns of type A blood, they use the lectin isolated from the cowpea to make those red blood cells, the blood of type A blood, agglutinate so that it can be used as a screening tool. And this is not new information in the area of hemodynamics and toxicology. It's only new really when I present this to clinicians of all types, and they start to understand that these lectins survive human digestion. They survive cooking. Boiling kidney beans for an extensive period of time really doesn't produce a significant drop-off in the lectin content of kidney beans. It's an example of a type of legume that just isn't that compatible with human physiology because of the lectin concentration. And when it really comes down to the communication that we're talking about here, all in the topic of inflammation, the inflammasome is upregulated with exposure to these agglutinators, or these lectins. Just like it is when certain bacterial populations are in greater quantities, there's an ability to downregulate that inflammasome. And again, the best examples would be your lactobacilli strains of gram-positive bacteria. Those are commonly found in yogurt. Rhamnosus is another excellent example of a bacteria which has been shown to downregulate the inflammasome, the inflammasome reaction. And when it comes to inducible heat shock proteins, our goal is to keep those upregulated so that we have a good, tight intestinal lining without that permeability. We have less inflammation. And similar families of bacteria that we know have better associations with most types of health, whether we're talking about digestive health or neurological health, it's really the same families of bacteria that we want to have larger populations of when it comes to upregulating. That's a good thing. Inducible heat shock proteins. Those are the Bifidobacter family. It's the lactobacilli, the Rhamnosus. These are also very beneficial when it comes to upregulating inducible heat shock proteins. So these lectins are a part of it. They're a part of the big picture here. But in every bit as important, or maybe even more critical component, is to having well diverse and well-represented families, important families of bacteria. But the research on lectins and a wide variety of chronic diseases that range from autoimmune type to neurological diseases is well established. And I think it should give all of us reason for many of our patients to consider a more carbohydrate-specific or low lectin, certain types of lectins at least, a lower lectin-containing diet. And again, that's not to say that all lectins do harm. We're going to talk really about the most damaging forms of lectins, and the two that are the most damaging are the lectins found in your large, broad beans, which would be kidney beans, fava beans, these larger beans, and wheat germ. The wheat germ agglutinin, WGA, is every bit
as destructive as the phytohemoagglutinator found in kidney beans. PHA and WGA are really the most damaging agglutinators that have been studied to date. And I would say that after looking at this research for the last 20 years, I feel very strongly that people do better without wheat germ and do much better without kidney beans, all people, than they do with them. It's really just common sense, I feel, at this point, to recommend diets, especially for people who have gut issues, high levels of inflammation. Recommend diets that contain neither of those. But here are some other, some other references that you may find interesting on this topic. Again, so the phytohemogglutinin and the wheat germ agglutinator, this is really the mechanisms that are involved, and it comes down to making white blood cells or red blood cells agglutinate. But as I mentioned earlier, not all lectins have the same overall effect. It's very interesting when you take a look at the lectins that are found in buckwheat, for instance, or the chickpea, or the garbanzo bean, I should say. These are examples of, well, although the buckwheat isn't really a grain, it's a dicot, so it's more a member of the vegetable family. And chickpeas are certainly legumes, but these would be examples of lectin-rich foods which appear to promote more favorable physiological responses because they do not stimulate the inflammasome the same way. They do not suppress the inducible heat shock proteins the same way. And so these would be examples of legume and grain-like based foods where there's much, much greater compatibility, and the observations made in a wide variety of investigations that range from ecological to small animal-based interventions would suggest that these are going to be much more compatible and much more healthy additions to a population's diet. So it's not to say that all lectins are damaging, it's just to say that we've got some with more dubious track records than others. Shifting gears, now looking at some of we've talked a little bit about gliadin, which again is the type of gluten that's been most researched, most heavily investigated by people like Alessio Fasano, and it's how it produces or stimulates zonulin production. We know that there are other less compatible proteins, and one of those that I speak about quite frequently when it comes to inflammation is the role of A1 beta casein, which is found in certain types of milk, most often found in modern varieties or intensively bred varieties of the cow. The Holstein cow, for instance, being the best example of the predominant source of A1 beta casein in the average American or North American diet, and that's because, of course, Holstein cows are the predominant cow in our dairy herd. Well, A1 beta casein is very, very different than A2 beta casein in terms of the way that the human body responds to it. But the differences molecularly are actually very subtle. They are only different in one amino acid, and it's way on down the protein chain. It's almost 70 amino acids into the protein complex. You have this one amino acid difference, but it produces such different responses in the body, and this has to do with the way that fragment, when it survives digestion, which it quite often does, and it enters circulation through a higher level of intestinal permeability, that fragment of casein from the A1 beta casein will stimulate a much, much different immune response by the human body than the A2 casein fragment, which is found in older varieties of cows, such as the Guernsey cow, but it's always goats and sheep milk, I should note, are always A2 casein types. The Guernsey is the only cow that is always A2 casein, so it's a really safe choice. But other varieties of cows would be highly dependent on their genetics. So Holstein cow milk, which is unfortunately the vast majority of milk in our food supply, if you go to a grocery store and just purchase milk with a blindfold on, it's almost always going to be A1 dominant or Holstein milk. But if you are selective with dairy products, it's becoming much easier to choose varieties that are predominantly or all A2, either using goat or sheep's milk products or now sourcing A2 tested milk. In Ohio, there's companies like the Snowville Creamery, which is all A2 milk. Out west in Colorado and California, it's become very easy to find A2 tested milk at the grocery store by a variety of companies. And in the Northeast, you could find it at a co-op or at a health food store if you were finding milk that said it was from Guernsey cows or Jersey cows that had been tested. So we're at a point now where people are becoming much more aware of the differences between A1 and A2 milk. And to be quite honest, when you hear associations made between a variety of dairy products and inflammation, gut issues, I think what's important for all of us to do is to take pause and consider the source of that dairy product. Is it predominantly A1 casein, or is it A2? And almost all of this research has been done that shows a link between inflammation or gut intolerance. Almost all of this research has been done with A1 casein. The research that's been done with A2 in areas like New Zealand and Australia shows a very, very different response to the human population's exposure to dairy products. A2 is much more compatible and much less inflammatory than A1 beta casein. So this is something I encourage all of you to look into in greater detail if you want to include dairy products in your patient's diet. It's not to say that dairy products have to be included, but they can be a great source of a variety of nutrients. It's important to understand the details. Now, I mentioned advanced glycosylated end products earlier as one of the 5 sources of increased intestinal permeability and gut wall or GI-based inflammation. And again, these advanced glycosylated end products can be formed in a variety of manners, whether that is because we have high concentrations of fructose in circulation. That would be a cause because fructose readily cross-reacts with proteins as well as other molecules to produce these age molecules, but it can certainly be produced in food processing with high heat cooking. It just requires 1 or 2 different substrates. It could require something that has an amine end chain. It could certainly be found in something that had a high fat or lipid content, and it can be found in things that have both a glucose content as well as a fat or a protein content. It really doesn't take very much carbohydrate to be present in a food brought to high heat. Even the small amount of carbohydrates found in the side chains of the different glycans found in the skin of chicken, for instance, when it's grilled, becomes a very, very rich source of these age molecules as it cross-reacts with the proteins and fats in that particular meat. But certainly heavily roasted nuts and seeds, heavily heated dairy products that have been ultra pasteurized and processed are going to be rich sources, besides the obvious sources, which would be browned and blackened meats, chicken and fish. Those would be very, very high sources. These molecules are very inflammatory to a variety of areas of our physiology, whether we're talking about the renal system or you're talking about the gut lining. They're a very significant source of inflammation. Some of the research with animals is fascinating to look at how much inflammation can be generated with a high-age diet and how quickly it can be attenuated or shut off when the age content of that animal's diet is immediately restricted. It really gives us, I think, just one more tool to work with our patients or the populations that we serve as a way to educate them. Low heat cooking, cooking foods in liquids or with acids are always going to be the much more healthy manners of preparing food. And it's going to protect our blood vessels and, as I mentioned earlier, our kidneys and the intestinal linings. Really a significant amount of research on age molecules and human health now that one could dive into. But it's just important to try to keep acid in the cooking system, whether that's something like tomato juice or tomato sauce, lemon juice, lime juice. These are all examples of acids that can stop, this age production. Cooking things in liquids is going to keep the temperature much lower as well. So it's always better to do that than to grill, brown, or blacken animal proteins eat more raw nuts and seeds. And that really brings us, in addition to getting the artificial ingredients out of a patient's diet, artificial colorings, artificial flavorings, preservatives, emulsifiers like carrageenan, for instance. All of these really need to go as we try to restore gut health and reduce inflammation. So that would be, again, that fifth category of dietary ingredients or compounds that can drive inflammation. And that brings us to our overall clinical pearls. How is it that we apply some of this information we've discussed today to reduce a patient's inflammation? That would take us to these most salient points. Eliminate wheat first and foremost, and it's not just because of gluten. And I think that's where we often see misinterpretations of a wheat-free diet. Is most of the investigation is on gluten. So some of these trials use isolated gluten as part of the investigation, and I think that using wheat germ agglutinator or WGA would be every bit as important or critical in terms of investigating the benefits of a wheat-free diet as would be gluten. So I think it's important to get rid of gluten and to get rid of wheat overall as a way to reduce inflammation and improve the gut environment. Eliminate soybean products and other large, broad bean or legumes that have high levels of PAH or phytohemoagglutinators. Avoid peanuts, even if a peanut allergy is undiagnosed, because there's a significant amount of lectins found in peanuts. They're a member of that broad bean family. Avoid all A1 beta casein containing dairy products and choose A2 types. Again, goat, sheep's milk, or Guernsey cow. We've talked about high heat applications. It's best to try to reduce the temperatures to if we can, under 300 Fahrenheit. 212 is going to be a much, much safer place to be when it comes to age formation. Eliminate any added sugar and flour-based products, as these are primary areas of developing dysbiosis. Of course, there are a lot of different ways that an individual could develop dysbiosis or particular bacterial or eukaryotic species overgrowth, but yeast, gram-negative bacteria, we've talked a little bit about Klebsiella, Citrobacter, for instance. These are bacteria that do really prefer very high carbohydrate, refined carbohydrate, in the case of Klebsiella, high amylopectin, starch-based diets. If we can reduce the amount of sugar and flour-based products in a patient's diet, that can go a long ways into supporting the changes that we want to make within their microbiome. Better choices. Choose organic rice or buckwheat whenever possible as better grains. In the case of buckwheat, a pseudo grain that will have more benefits and less drawbacks than members of the wheat or barley family. Choose organic lentils, smaller legumes like the aduki or adzuki bean or chickpeas. Choose these types of beans or lentils more often as the legume, and try to use extensive cooking and heating times, meaning that they're simmering or boiling for a couple hours as opposed to just 30 minutes. This can help. This type of extensive cooking and or soaking fermentation process can significantly reduce the lectin content. Choose organic raw nuts and seeds over those which have been heavily heated or roasted. I've talked about cooking meats at lower temperatures and using acidic juices or the juices of vegetables. Making stews and soups is really the way to go. And limit sugar to those which are, naturally occurring in fruits. And for the fruits that we eat most often, it's always best to choose fruits that have the most upsides with polyphenolic contents and more acids and less in the way of fructose. Those would be berries and tomatoes, along with members of the citrus family. And so with that, I leave you with the herb of remembrance, rosemary, and hopefully we can remember some of this. And now what I'd like to do, the last 10 minutes that we have, is answer what questions that you may have.
John, if you go to the questions tab, you'll see a list. Just
Right.
I can read them too if that's easier.
Yeah, sure. I will. I'll start out with. Well, let's see. Is a lot of it has to do with the noise. I really apologize for the scratchy mic.
Oh yeah,
I
I've
guess that went on
I've
before.
cleared those, so I have a list here. I'll read them to you.
Thank you, Catherine.
Okay. Which metabolites feed pathogenic bacteria? What food are they in?
Sure. Well, that's a great question. And the metabolites that are going to be more of a problem for an individual with dysbiosis, those metabolites are going to primarily, that you want to look at closely, those are going to be a function of the pathogenic bacteria that is playing a role in a person's autoimmune or inflammatory based condition. So I gave a couple examples. One that we know really well is, again, Klebsiella. That's why the London diet that was developed well over a decade ago now has been so effective in helping people with ankylosing spondylitis. But another example would be the Shigella toxin or Shigella family of E. coli that many people have exposure to. That is a really interesting microbe because when people have diets higher in red meat and or dairy, that particular microbe can take the sialic acid NEU5GC found really only in red meat and dairy products, and it can incorporate that into our own tissues, and that will stimulate autoimmune attack of those particular tissues. And it's been shown with Crohn's disease as well as with Hashimoto's, again, an autoimmune disease of the thyroid. So I can't say that there's a particular dietary metabolite for, that's going to feed all of these families that can drive autoimmune. It is highly dependent on the family of bacteria, but really sugar, and I think most of us know that, sugar, refined carbohydrates, refined starches tend to drive more types of dysbiosis in the human microbiome than any other family of foods or groups of foods. So that would be my starting point to answer this question that, as best I can. If there was a good starting point, it would be to eliminate added sugar, refined carbohydrates, refined starches that are going to be, most often feeding particular patterns of dysbiosis.
Can you share the specific bacteria associated with Hashimoto's?
Sure, that one example is the Shigella bacteria that I just mentioned.
Okay. What is your take on oil intake on inflammation, such as olive oil, for example?
Yeah, this is something I've looked at really closely for a variety of reasons. Once in my clinical practice, I, used to recommend that people would use more fish oil, and I now do not recommend that. Every year, I try to take a very, very close look at a variety of oils and the role that they play on the inflammasome and on intestinal permeability and overall health. And I'm at a point now where I think extra virgin olive oil and organic flax oil are the two liquid oils that I feel most confident in as being great additions for less inflammation and not causing any inflammation. I also think many people can do very, very well with, these are not liquid oils, but coconut oil, grass-fed butter. These are solid fats or lipids. But I think it's really important to limit the oil in a person or patient's diet to high-quality extra virgin olive oil, which has been shown to, for everything we've talked about this morning, produce favorable effects, whether it's inducible heat shock proteins, the inflammasome, really reducing inflammation. Extra virgin olive oil, organic flax oil are my two top choices, and I think they have anti-inflammatory properties in addition to being very well tolerated or not producing inflammation in any way.
So a few questions about lectins. I'm going to just kind of tell them also. There's a question about lectins in black beans. Can we correlate the amount of lectin in a food to be more damaging? examples of beans, legumes with positive lectin profiles.
Yeah. So is that a good starting point, Catherine?
Yes, and then just how the role of glyphosate as well with lectins on chickpeas and legumes.
Sure. So I'll start off with just the broad topic of lectins. And again, I mentioned this in the talk, but lectins are a very, very broad category of plant defense proteins. These are substances produced by plants to defend themselves against a variety of pests. Now, they're not all damaging to the human gut lining, but some are, and some also have the potential to cause our red blood cells or our white blood cells to be less effective and to cause abnormality within that area of physiology that we call hemodynamics. That just has really looking at what are the different attributes of our blood and our blood's ability to defend ourselves, to carry oxygen, and to help circulate everything needed by the body. And once you start to disrupt red blood cell function and white blood cell function, you're really opening up a significant potential here for suboptimal human health. So I don't think it's good for us to consume those substances that have good amounts of clinical evidence that their lectins, this would be the kidney bean, the fava bean, the jack bean, the broad bean, these are big, broad beans with notable hemagglutinator. These are lectins that cause damage to various blood cell types. I recommend avoiding these rich sources of lectins, but I do not at this point in time have any clinical evidence that one should avoid chickpeas, small red beans like the aduki bean, or lentils. Even the black bean, I would say it would be circumspect to recommend avoiding black beans because I don't have enough damaging evidence on the lectins contained in black beans. But I do with kidney beans and the big broad beans. And this probably has a lot to do with the amount of energy and food that's available in one of these big pods that sits in the ground for a period of time. It's going to be desired by a variety of pests in that environment. So that plant's going to really have to defend itself more aggressively than, let's say, a lentil, which has a shorter germination time. It spends, it doesn't have the available carbohydrate, etc. So not to get too techie here with the botany, but I don't have the evidence, and I have some evidence that people who eat chickpeas are more nourished. That's a wonderful paper. It was a few years old now that was done in the UK, and it looked at populations that had varying levels of chickpeas in their diet, and those that had more chickpeas in their diet tend to have higher biomarkers that indicated good levels of nourishment, better overall health. So, when it comes to chickpeas and lentils, I generally have only seen good things in the literature. They do contain lectins, but those lectins do not appear to cause the same type of damage to the gut lining, to our various aspects of our blood that the big beans do. So that, I hope, can answer that question. That I don't think people need to avoid lentils, chickpeas, or the smaller legumes because there is no evidence. There is evidence, though, with the kidney bean in particular that it's damaging. And I also recommend that people avoid members of the wheat family because of the lectins contained there. But I don't recommend that people avoid rice or buckwheat, for that matter, because they do not have lectins with damaging effects to the human GI. So that would be my long and the short on lectins, and something that I will continue to investigate at least monthly, as I always review any new paper that's come through in this area of food science. Now, the last part of that question had to do with glyphosate. Glyphosate, what I do know about glyphosate is, as an herbicide, it damages the shikimate pathway, and that rendering that pathway or incapacitating that prevents a bacteria from producing important benzene or ringed amino acids, aromatic amino acids like tyrosine, tryptophan, and phenylalanine, and that will really limit the population of important families of bacteria, which are very anti-inflammatory and that play pivotal roles in outcompeting pathogens. And so glyphosate suppresses bifidobacter populations, and bifidobacter populations are really important for the intestinal environment, and they outcompete Clostridia, for instance, which is a that's an example of dysbiosis that we really all want to avoid, and that's an example of why we want to avoid high sugar and refined carbohydrate-based diets, because Clostridia really feeds on those metabolites. So, I would say that in summary, glyphosate damages the microbiome by eliminating bifidobacter populations in individuals with glyphosate exposure, and that's going to make them more susceptible to particular pathogen overgrowths.
Okay. What about the use of digestive enzymes to support digestion of some of these problematic compounds?
Absolutely. I think people can really improve the gut environment with a combination of digestive enzymes. Some individuals require more acid support in the upper GI. We have a large number of people that have very, very low levels of gastric hydrochloric acid production. That's going to compromise gut health. So I think digestive enzymes is a great tool for clinicians to use for people who have high levels of gut-based inflammation, and it's either demonstrated or suspected that there's a high level of permeability there. I would definitely recommend a digestive enzyme that contains significant amounts of particular proteases, which are often called DPP. These are dipeptide proteases, like DPP Roman numeral 4. That's a very beneficial one for people who have had prolonged gut wall damage from exposure to certain lectins or gliadin. But I think they can go a long ways, that in combination with a probiotic or the right fermented food as well.
People who live in blue zones seem to have plant-based diets with grains and olive oil. How do you explain this?
I don't know as though I need to explain that. That's something that people always bring up. And I would say, let's take a look at those blue zones diet. I know them very well. I've spent time in most of them. The Nicoyans, for instance, on the Nicoya Peninsula of Costa Rica, they ate a significant amount of beef. I'm not sure if people are aware of that. There are plants in their diet, and you might still say that it's plant-based because all of the beef is grass-fed. But they eat some black beans. They eat a significant amount of beef. They get a lot of sunlight. They're in a beautiful climate overall. The Sardinians eat small amounts of wheat that is made into very, very thin flatbreads, and then folded into that is a copious amount of cheese and olive oil, as this person mentions, and an abundance of locally grown fruits and vegetables, as well as a wide variety of animal products, meats. I wouldn't say that the Sardinian diet, from spending time there in the early 1990s, I wouldn't say that that is a diet that would be plant-based by definition. I think that would be very misleading. The Okinawans, of course, eat a variety of sweet potatoes throughout the year, a variety of rice, but the Okinawans don't eat wheat, and they don't really eat significant quantities of lectin-rich foods. So we could go through these in great detail, and I will say that these blue zones do have some of these things in their diet in varying amounts, but it's not like the amount of wheat, for instance, that you'd find in the American diet. It's not the amount of A1 beta casein that you find in a North American diet. None of these populations eat kidney beans. So they may have varying levels of plants in them, and I'm not here to in any way devalue a plant-based diet. I just think it's really important to understand that not all plants are benign or beneficial. I think it's really important for people that are advocates for a plant-based diet to start to acknowledge that certain plants, whether we're talking about lectins or plants that carry high levels of agrochemicals, like soy, for instance, that we start looking at things like wheat and soy and heavily agrochemical contaminated plants as not being integral components to a plant-based diet, but things that we probably want to be a little more vigilant around. And I would argue that the best plant-based diets would be those that are rich in vegetables and are rich in animal products that are completely plant-based, where the animal is entirely grass-fed. So, I don't think that the blue zones, in summary, are in any way evidence to anything of the contrary that we've talked about today. I would say that when you look at the blue zones, most of them have very strong communities. They tend to enjoy meals with one another. And look, I mean, in the grand scheme of things, when we take a look at the role of nutrition in influencing human health, I think it's really important that people share meals together, that they enjoy food, that we don't experience anxiety or paralysis around food. One of the things that I always feel conflicted around is that I try to share information. I really just try to share the facts. I'm not here to promote any particular way of eating, despite how it may sound, other than I try to share the science that I have become exposed to, educated around, and whether someone chooses to be vegan or entirely plant-based or more paleo, I think that's up to the person, but I think that there are a wide variety of choices that we can make under any of these ways of eating. And it's good to get rid of those foods or eliminate those foods that are less compatible, that are not sustainable to the planet's environment, and that just really don't make that much sense on many levels. But one thing that when I was going with is that I really cringe at the fact that we, unfortunately, experience some type of isolationism by the way we eat. I think it's really important when we look at those blue zones, we think about the role of nutrition in our health, that we let food bring us together. Either that we're enjoying meals together, or that we're coming together as a global community as a way to stand up for a population's rights or a particular aspect of environmental health that's being threatened by certain types of agriculture. And so with that, our time is up, and I really appreciate your attention this morning. I wish all of you the best and look forward to talking to you in the future. Thank you for being part of this webinar.